Thank you, Baxley. Could I share my slides? Firstly, have a look at that. Okay.
See, I can see.
Okay, thank you. I'm Manabato, and nice to see you, everyone. And I would like to briefly introduce about our research center and also the today's speakers, the Dr. Yumiko Tsikamoto, senior researcher in our centers. So let me briefly talking about our Leprosy Research Centers, which is established in 1955 as an affiliate of the Ministry of Health and Welfare in Japan. And the name has been changed in 1997 to a Leprosy Research Center of National Institute of Infectious Diseases. And we originally, our center is originally established as a research center for leprosy, but as you know, leprosy is a very difficult disease, and this causative bacteria is not culturable. That's why we expanded its research area into other mycobacterium diseases, such as tuberculosis and non-tuberculosis mycobacterial diseases as well. And so we, in 2008, we established the BSL-3 and ABSL-3 laboratories, as shown in the right photos. So we started the vaccine development researches, which is today's subject, maybe spoken by Yumiko. So Dr. Yumiko Tsikamoto belongs to laboratory number 5 in our research institute. And also she have been working for development and the researches for recombinant BCGs for more than a decade. This recombinant BCG expresses bacterial protein, which is called MMP-II. And also this protein is expressing in both in Mycobacterium leprae and Mycobacterium tuberculosis. That's why we expected this vaccine could prevent and it could be effective for TB and leprosy in the both. So that's it for my presentations, and also I pass to Yumiko. If so, are you ready for? Could you share your slide, please?
Thank you, Chair and Dr. Ato. I'll share my slide.
Yes, it's shared to everybody.
Okay, and okay, and can you see pointer now?
We can see the points.
Oh, okay. So I'll start the presentation. And I'll talk about our project, development of recombinant BCG vaccine for tuberculosis utilizing messenger membrane protein 2 antigen. And first, I'll talk about current situation of tuberculosis briefly. Infectious disease caused by Mycobacterium tuberculosis. Estimated global TB incidence is 10.6 million, and TB death is 1.6 million each. And this map shows the countries where many incidents were detected in 2021. As you can see here, in South and Southeast Asia and Africa, there are many incidents of tuberculosis. And this slide shows the annual number of deaths and TB incidents over time. As you can see here, annual number of deaths by TB is increasing in 2021. In 2021, TB was the second leading infectious killer after COVID-19. And the COVID-19 pandemic has reversed years of progress made in the fight to NTB. So, to NTB, efficient vaccine will be required. To fight TB, BCG has been the only vaccine against tuberculosis. BCG is cost-effective and safe vaccine. It is very good. But the efficacy of BCG is controversial. In this recent paper, it has been shown that BCG is effective in children, especially under 5 years old. However, BCG is less effective in adolescents and adults. So why BCG is less effective in adults? This slide shows our hypothesis. It may be because insufficient memory T cells. If this hypothesis is true, we will be able to develop more efficient vaccine by improving the naive T cell activation by BCG. And from this hypothesis, we have developed recombinant BCG, which can activate naive T cells efficiently. And the key antigen of our recombinant BCG is MMP-II. It means major membrane protein 2. And major membrane protein 2 was first identified from Mycobacterium leprae because our institute has been mainly focused on the research of Mycobacterium leprae. As the immunant-dominant antigen, we identified major membrane protein 2. And fractionation of Mycobacterium leprae membrane proteins revealed that MMP-II stimulated interferon gamma production from T cells. MMP-II is a membrane protein, and it is also a TLR2 ligand. This graph shows the luciferase reporter assay using TLR2 transfected hex cells. MMP-II activated NF-kappa B in TLR2 dependent manner. As positive control, peptidoglycan was used. And we have developed MMP-II expressed recombinant BCG, and protection assay against Mycobacterium leprae was conducted using black 6 mice. BCG was vaccinated intradermally, and 4 weeks later, Mycobacterium leprae was inoculated in the foot pad, and 7 months later, bacilli in the foot pad were recovered and counted by Thiel-Nielsen stain. And these results indicate that MMP-II expressing BCG was more effective in inhibiting Mycobacterium leprae multiplication than control BCG. So our question is here. Can MMP-II expressing recombinant BCG also inhibit multiplication of Mycobacterium tuberculosis? Here is the homology of MMP-II from Mycobacterium leprae, Mycobacterium tuberculosis, and Mycobacterium bovis BCG. MMP-II from leprae and MMP-II from TB has 90.6% homology in amino acid level, and MMP-II from MTB and MMP-II from BCG is totally identical. We had already shown that MMP-II from leprae has strong immunogenicity. Then what about MMP-II from TB? To answer this question, we have purified recombinant antigens from Mycobacterium smegmatis. Recombinant protein expression plasmids were introduced into smegmatis, and those 4 recombinant proteins were purified. Then we compare the immunogenicity. The purifiers are MMP-II from Mycobacterium leprae, MMP-II from Mycobacterium tuberculosis, and fusion protein of HSP70 and MMP-II from Mycobacterium leprae. We call this fusion protein as fusion ML. And the last one is the fusion protein of HSP70 and MMP-II from Mycobacterium tuberculosis. We call this fusion protein as fusion MTB. And now you must be wondering, why does HSP70 appear suddenly? HSP70 is one of heat shock proteins in TB and BCG, and it is secreted from BCG. So the fusion protein will be secreted from BCG. And HSP70 is well known that it has potent adjuvant activity. Therefore, we examine whether HSP70 MMP-II fusion protein has strong immunogenicity. First, we stimulated human disease with those recombinant proteins in vitro. When MMP-II from Mycobacterium leprae and MMP-II from MTB were compared, MMP-II from MTB induced more IL-12 production from human disease. And when fusion ML, which is fusion protein of HSP70 and MMP-II from Mycobacterium leprae, and fusion MTB, which is fusion protein of HSP70 and MMP-II from MTB were compared, fusion MTB was more immunogenic. And from this experiment, you can see that fusion MTB was the most immunogenic protein. And next, we analyze the activation marker on dendritic cells stimulated with MMP-II from MTB or fusion MTB. And you can see here, fusion MTB promoted expression of surface antigen markers than MMP-II alone, surface antigen markers and HLA class 1 and class 2 antigens. Then we pulse disease with those recombinant proteins and activated naive CD4 positive T cells. As you can see here, fusion MTB promoted interferon gamma production from T cells efficiently. We next analyze using CD8 positive T cells, and again, fusion MTB was most immunogenic. And this slide shows the perforin expression of CD8 positive T cells. Fusion MTB stimulated perforin expression of CD8 positive T cells efficiently. So here is the summary of the in vitro experiments. MMP-II from MTB and fusion MTB, which is fusion protein of HSP70 and MMP-II from TB, can efficiently activate DC and promote T cell activation. And the immunostimulatory activity of the recombinant proteins examined is here. As you can see here, fusion MTB is the most strongest of those 4 proteins. Therefore, we should make recombinant BCG expressing fusion MTB, which is HSP70 MMP-II fusion protein. And there is another strategy of improving BCG vaccine. BCG has the major intrinsic defect. When BCG infected phagosome-lysosome fusion is lacked. It leads to weak presentation of BCG-derived antigen, and then insufficient stimulation of T cells. In order to overcome this defect, we produced urease deficient BCG. UreC gene was deleted from BCG so that no ammonia was produced, and therefore the acidification of phagosome was possible. We named this urease deficient BCG as BCG delta UT. And BCG delta UT activated human disease and enhanced expression of class 1 HLA class 1 and 2 molecules and activation markers on human disease than parent BCG. Also, human naive T cells were efficiently activated by BCG delta UT infected disease. So, here is our first recombinant BCG against tuberculosis. As host BCG, we use BCG Tokyo strain because BCG Tokyo strain seems to be the most safest strain of BCGs. And this recombinant BCG, UreC gene was depleted, and fusion MTB is expressed by plasmid introduction. This UreC gene depletion leads to acidification of phagosome and secretion of fusion MTB. Secretion of fusion MTB and acidification of phagosome, when both are combined, improved vaccine efficacy against MTB may be attained. We named this recombinant BCG as BCG DHTM. And we confirmed the expression of HSP70 MMP-II protein by Western blotting. First, we compare the immunostimulatory activity of BCG DHTM and control BCG in vitro. When human disease were infected with control BCG or BCG DHTM, BCG DHTM efficiently induced activation markers on disease. And here is the result of cytokine production by disease infected with BCG. As you can see here, out of B70, TNF-alpha, IL-1 beta, BCG DHTM promoted cytokine production more efficiently than control BCG. And when human naive CD4 positive T cells were stimulated with BCG pulse disease, BCG DHTM induced production of interferon gamma by T cells more efficiently than control BCG. And this production was inhibited by anti-HLA antibody and anti-CD86 antibody. And when human naive CD8 positive T cells were stimulated with BCG pulse disease, BCG DHTM induced production of interferon gamma by T cells more efficiently than control BCG. And this production was inhibited by anti-HLA antibody and anti-CD86 antibody. We also examined perforin expression of CD8 positive C cells stimulated with BCG pulse disease. BCG DHTM induced perforin expression more efficiently than control BCG. And finally, we have examined the vaccine efficacy of BCG using mice model. Black 6 mice were vaccinated with control BCG or BCG DHTM, and 6 weeks later, mice were challenged with Mycobacterium tuberculosis 837RV. And after 6 weeks, mice were sacrificed and Mycobacterium tuberculosis CFU was analyzed by CFU assay. And the result is here. BCG DHTM more efficiently inhibited the multiplication of Mycobacterium tuberculosis in vivo. Then we tried another improvement. We introduced PEST sequence into the both ends of the fusion protein. PEST sequence is a signal peptide characterized by 4 amino acids P, E, S, and T, and associated with processing of various proteins. So by introducing PEST sequence, we expect more efficient T cells activation and improvement of recombinant BCG. And here is the result. We compare the recombinant BCG having PEST sequence with control BCG and BCG DHTM. As you can see here, BCG PEST is more efficient than control BCG and BCG DHTM. So this is the summary of BCG DHTM and BCG PEST. Both BCG DHTM and BCG PEST have higher vaccine efficacy against tuberculosis than control BCG. And those recombinant BCGs are hopeful as new vaccine against tuberculosis. However, these recombinant BCG have hygromycin-resistant gene, and antibiotics-resistant gene must be depleted before practical use. And to deplete antibiotics-resistant gene from our recombinant BCG, we utilize auxotrophy. We depleted loaded gene from BCG, and this loaded gene is essential to synthesize lysine. In loaded deficient BCG, loaded containing plasmid is maintained without antibiotics. So we developed recombinant BCG, which is antibiotics-resistant free, expressing PEST HSP70 MMP-II fusion protein and depleted with UreC gene. We named this recombinant BCG as OXO-BCG and examined the vaccine efficacy of this BCG. And here is the result. OXO-BCG is more efficient than control BCG. And the efficacy is similar as BCG PEST. So this is the conclusion of our study. UreC gene depletion and HSP70 MMP-II fusion protein secretion together enhance the vaccine efficacy of BCG. This recombinant BCG is BCG DHTM. And introduction of PEST sequence into HSP70 MMP-II fusion protein also enhance the vaccine efficacy. This recombinant BCG is BCG PEST. And recombinant BCG can be made antibiotics-resistant gene-free by utilizing loaded depletion. This is OXO-BCG. And this is the future prospect. We developed recombinant BCG vaccine. Those vaccines based on BCG may be easy to implement due to safety issues compared to other lab-attenuated vaccines. And our recombinant BCG vaccine may be applicable to tuberculosis as well as leprosy prevention because both bacteria express MMP-II. And as you have seen, MMP-II antigen is very immunogenic. So MMP-II protein may be useful as booster vaccine with appropriate adjuvant. If you have very good adjuvant, we hope you will tell us. We will greatly appreciate if we can test the combination of MMP-II and your adjuvant. And this slide shows the collaborators at Leprosy Research Center of this study. Thank you for listening.
Thank you, Yumiko. So this paper is now open for question and comments from the audience. Is anybody who have a comment, any comment and questions? Please, I think off the mute and then ask the questions about our vaccine. Is anybody? Okay, one I have. Okay, thank you. Thank you. Yeah.
Thank you.
Well, you're welcome. Any question and comment?
Feel free to type your question if you're shy, but do come online and ask any questions or comments.
And also, just making comments about future progress of the design of recombinant BCG, because this talk, Yumiko shows that plasmids incorporated recombinant BCGs, but this kind of incorporation of plasmids could be excluded by a BCG during the long time immunizations. So we already established to chromosome incorporated recombinant genes into the BCGs. So this could be also be very stable for more than 1 year for keeping recombinant genes, and also the express nice immune capacities. So I think that we now kind of chromosome incorporated BCG, recombinant BCG for preclinical studies, and also the perhaps in phase 1 in the futures. Okay, so can I read? Raise your hand. Any comment and questions?
There's a question in the chat, Manabu.
Yes. Are you planning to do any safety studies in skid mice? So, Yumiko, do you plan for the safety studies using skid mice, especially in terms of the billions of recombinant BCGs?
Yes. Yes. We are planning to do safety studies in using skid mice, and our recombinant BCG, the host is BCG Tokyo strain, and BCG Tokyo strain is quite safe, even in skid mice. So we expect that our recombinant BCG is also safe in skid mice.
Okay, thank you. So I think that the Tokyo 172 is quite different from Danish strains or the other pastoral study, because this could be somewhat, I think it has been reported about BCG substrain has different immunogenicity and also the safety. So any other questions or comments? Are you planning on looking at whether the recombinant BCG can be used as a booster BCG? BCG is still given to the infant in a lot of countries. Yumiko, can you answer the questions?
Yes. We are also planning the recombinant BCG can be used as a booster vaccine. Yes, as the Jessica says, BCG is very effective in infants, and recombinant BCG as a booster vaccine may be useful, we think.
Yeah, so thank you for Jessica. Thank you for your questions. And I think Yumiko already reported. I think there are at least two options. One is a recombinant BCG using as a booster, but because most of children receive the jab of BCGs in when they have they are infant. But we also have any plans. So if our recommended BCG is much better than the classical conventional BCG, we could plan to replace this jabs of BCG into the recombinant BCG. And also we have started combination studies for using recombinant BCG as a primary vaccine, and also the boosting for recombinant protein vaccine candidates. So to see whether these combinations could show more protective immunity against tuberculosis, which could, because lots of vaccine candidates fail or not shown not for efficacy as we expected. That's why I'm thinking our recommended BCGs could make a kind of a breakthrough for this kind of immunogenicity and also efficacy and in terms of TB vaccine programs.